In eukaryotic cells, Krebs cycle happens in the matrix of the mitochondrion under aerobic conditions, which means that this pathway requires oxygen. One single cycle produces 1 ATP, 1 FADH2, 2 CO2, and 3 NADH. NADH is used in the oxidative phosphorylation later. In glycolysis, 1 glucose molecules produce 2 acyl-CoAs, which means that you have to double these numbers if you want to know the products of 1 glucose molecule. There are 8 steps in the cycle. First molecule needed is acetyl coenzyme A from the glycolysis. We will start with the molecules. You can use either the name of the acid or the salt form. The molecules are citrate. The word reminds me of citrus, so there will be a lemon. Isocitrate. The word iso means equal. I add a lime since it is equally also a citrus fruit. Alpha-ketoglutarate. You can also call this one alpha-Kg. Kg reminds me of the word king. Succinyl-CoA. Succinyl sounds a bit similar to the word success, which the cup represents. Succinate is similar word as earlier. Let's add mouth for the ending 8. Fumarate. The first thing that comes to my mind is the movie Fast and Furious. And I will add some flames to underline the word furious. Malate. No need to think, this will be a manatee. Oxaloacetate will be an ox with an acetate tail. Now you can try to make up a story to remember these molecules. The more stupid it is, the more likely you will remember it. For example, there was a lemon and lime. A lemon wanted to be a king. It was dreaming about success. Unfortunately, somebody came and ate it. A lime became so furious that it drove its car into an ocean, where manatees were swimming around and an ox watching the show from the waterfront. The next step will be the enzymes. I will choose color for each of them, so it will be easier to figure them out. There will be 4 dehydrogenases, 2 synthetases, 2 others. Let's place first the others. They are aconitase and fumarase. Aconitase you can remember from the artist Akon. Fumarase converts fumarate into malate. Then we will add synthetases, which are citrate synthetase and succinyl-CoA synthetase. As you may notice, the rule in the Krebs cycle is that the name of the enzymes comes from the molecule that it is about to convert. There are 2 exceptions: citrate synthetase and aconitase. Since we are now left only with dehydrogenases, it is easy to fill them in. These are isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, succinate dehydrogenase, and malate dehydrogenase. From one cycle, we will get 1 ATP, 1 FADH2, 2 CO2, and 3 NADH. Find the 4 hydrogenase enzymes of the cycle. From these steps, we will receive NADH, except from the reaction, where succinate dehydrogenases work. This will give us FADH2. First 2 of them will also give away CO2. There is one lonely step between these 2, so we will add GTP there. It will be converted into ATP. Now you just need to rehearse the cycle by drawing it by yourself. I recommend that you do it this way. 1. Try to remember how many steps there are. Fill all the molecules. 2. Try to remember which kind of enzymes there are. Fill first others, then synthetases. Now there are only dehydrogenases left. 3. Lastly, add coenzymes. Search all the dehydrogenases. First, fill FADH2, the rest of them will be NADs. CO2 comes from 2 first of the dehydrogenases. The succinyl-CoA synthetase step is now lonely, so add GTP there, and remember that it will be converted to ATP. Thank you for watching. Comment on which pathway you would like me to do next.